nous allons passer au prix Dubois-Brigué 2020 pour la pathologie tropicale. Je ne sais pas si le professeur VERCRUYSSE ou Étienne Pays sont dans la salle. Sinon, je vais lire la délibération du jury. Donc, c'est en fait le docteur Caljon qui a obtenu le prix. Et Caljon, as only candidate, received the prize for his work on control of sleeping sickness and Leishmaniasis from insect bite to effective treatment for submission. His work concentrates on the treatment against sleeping sickness and Leishmaniosis and the role of innate immune responses on the pathological outcome of the disease. Novel insights have been gained in the early immunological processes that underlie successful systemic colonization of the mammalian host following intradermal inoculation of trypanosomes by the infective bite of a tsetse fly. These recent findings revealed that parasites can co-opt host innate immune responses, neutrophil function for enhanced infection establishment. Conversely, in Leishmaniosis, research on the only oral antileishmanial drug, miltefosine, resulted in unraveling the mechanisms of resistance that is linked to defective drug uptake. Very surprisingly, resistance parasites were found to significantly benefit from exposure to the drug, which strongly advocates for discontinued use of miltefosine in areas where treatment failure is on the rise. Continued research is aimed at obtaining more detailed information about the mechanism of action and drug targets. The antileishmanial leads are now further tested in combination treatments for move towards clinical evaluation in the very near future. For the antitrypanosomal leads, there appears no immediate commitment to a new human African trypanosomes drug development program, but the discovered lead series will further be explored for African animal trypanosomes. Je vais céder la parole alors au docteur Caljon.
So thank you very much for the introduction, and so good morning, Mr. President, board members of the Belgian Royal Academy of Medicine, dear family members and colleagues. It's a pleasure and privilege to receive this Dubois-Brigué Prize, which highlights our research on sleeping sickness and visceral leishmaniasis. These are actually two parasitic diseases that are lethal and are transmitted by blood-feeding insects. For sleeping sickness, this is the tsetse fly. And for visceral leishmaniasis, these are the sandflies. Now, in my research team at the Laboratory of Microbiology, Parasitology and Hygiene at the University of Antwerp, our research scope deals with both aspects ranging from understanding the early onset of an infection following an infectious bite towards contribution to novel treatment therapies. So one of the research questions we have is, why are these parasites so efficiently transmitted from one host to another? And that immediately highlights actually the importance of the insect vector. Now, in order to answer questions related to transmission, of course, you need the insect vector, and for this we decided that it's essential to establish an insectary at the University of Antwerp. And so we have now an insectary where we raise or rear both sandflies and tsetse flies, and we are able to infect them and use those to conduct natural infection studies, which are important to understand these early processes. Now, for parasites to be transmitted, they have to go through several bottlenecks. They have to be able to establish an infection in the midgut. They have to move upstream and eventually differentiate into infective forms that can infect, again, a new host. So what we have done is we have compared many strains with either natural resistance or induced resistance against existing drugs or new drugs to anticipate the risk of spread of the resistance. And so we have seen that depending on the resistant mechanisms, some parasite strains are very efficiently transmitted. So this of course implies a number of risks related to transmission and acquiring resistant parasites. Now another aspect we are highly interested in is because currently there's no human vaccine that can protect humans from infection, is to understand the early infection onset and how these parasites can escape the innate immune response. And so for this we do infections with reporter parasites. They express fluorescent or bioluminescent genes, and we can track infection in these mice, which are transgenic mice or gene-deficient mouse models. And quite unexpectedly, we have seen that a major innate immune cell subset that has been dealt with before in one of the presentations as well, the neutrophils. Well, rather than protecting the host from infection, these neutrophils seem to contribute to infection, which means that these parasites not only have gained resistance against the effects of neutrophils, they really have co-evolved with the host to make use of the mechanisms of the neutrophil. So this definitely gives us new insights in the early infection establishment by these parasites. Now, another major aspect or problem in treatment, and especially in treatment of visual leishmaniasis, is the problem of treatment failure. And now one of the most normal mechanisms of treatment failure is that the parasite acquires a drug-resistant phenotype. And so we have studied many mechanisms of resistance against existing and new drugs. And one example is where a single mutation, of course, can give rise to resistance by a mutation in the drug transporter. So as a result, these parasites no longer take up the drug and hence become fully resistant to the drug, and also they're highly transmissible. So that's an important risk for the human population. Now, something which is a hot topic now in the lab is that we have seen that Leishmania parasites also can persist in sanctuary cells in the bone marrow. And so we have immunophenotyped these cells. These are long-term hematopoietic stem cells. And we have seen that these cells are extremely vulnerable to the parasite infection, actually much more vulnerable. And these dots that you see, these are the intracellular amasticos, the Leishmania parasite. You can see that these cells are much more susceptible than the macrophages, which are known to be the major host cell in the host. So also these cells are very difficult to treat. So it's very difficult to eliminate these parasites from these host cells. And based on studies on the mechanisms, of course, this gives us mechanistic insights in treatment failure as well as in relapse. Now, finally, given the medical need, we also have contributed to the development of novel treatments, as we are the reference screening laboratory for the Drugs for Neglected Disease initiative, also called DNDi. And based on our in vitro and in vivo studies, we have actually moved forward 3 novel antileishmanial drug classes, the nitroimidazoles, the oxabaroles, and the aminopyrazoles, from which 2 are currently in phase 1 clinical trials. Also in combination or in collaboration with the University of Ghent, we have identified highly potent nucleoside analogues that are highly effective against sleeping sickness. And they are actually also active across the blood-brain barrier, which is an essential aspect of treatment for sleeping sickness. This has been patented. Now, last but not least, we also have developed a new diagnostic tool for visual leishmaniasis. This is based on the molecular detection of a small RNA molecule, and this test outperforms actually the gold standard, and it also provides a better test of cure. So we had the opportunity to install this test in the Research and Treatment Center in Gondar in Ethiopia, where actually the following clinical trials will take place. So I would like to end here by thanking my research group at the University of Antwerp, the funding agencies, as well as several collaborators, my family, and of course the Dubois Foundation for this honorable recognition. Thank you.